we've been talking about cellular respiration, and before we go into any more details, let's talk about what we already know. What can you tell me about what we've discussed so far? Please and thank you. Glycolysis is one of the steps of the process we're studying. Yes? What else have we talked about overall? There's 4 steps. What 4 steps in what? In creating ATP, which is what we call cellular respiration. That's the name of the process. Cellular respiration, we're creating ATP. Big picture, what are we using to create ATP? Food. Yes, food. So food goes in, ATP comes out. The respiration part is because oxygen comes in and carbon dioxide is left out. So respiration is O2 and CO2. And then cellular respiration, that's the name of the process of taking food and turning it into ATP. But the respiration part is because oxygen does go in, carbon dioxide does come out. What else do you know that we had talked about the last time we were together? Okay, fantastic. So every time we run through all 4 steps, and we're going to kind of finalize all that today, you generate around 36 to 38 units or molecules of ATP. Very good. What about generally this thing right here? It is a mitochondria. Fantastic. Can you tell me anything that's unique about this organelle? Why does it have its own genetic material? You are exactly right, it does, but why? It used to be its own organism. More specifically, it was a bacteria. Back in the day, like a long day. Okay? Besides it having its own genetic material and us knowing that it survived on its own, what other physical characteristic does it have that also reinforces the fact that it once was outside of a regular cell? It has 2 cell membranes. Why does it have 2 cell membranes? Exactly. When it was a bacteria by itself, it had 1, right? And then how did it get inside the other cells? It was engulfed, and we learned the name of that process. The name of the process of bringing something into a cell is called endocytosis. And more specifically, when you bring large things in, we call it phagocytosis. So when you do that and you bring it inside, you create it starts with a V. Close. Vesicle, yes. So the cell membrane, remember, it kind of folds around it, and it creates a vesicle. And because it's your cell membrane that's engulfing it, it's a phospholipid bilayer. So this bacteria that was all by itself, when it was engulfed through the process of endocytosis, more specifically phagocytosis, it had a vesicle around it. So the reason it has 2 cell membranes is, one, it belonged to itself, and the second one was remnants of the vesicle that brought it in. Those are unique characteristics that we only find in this organelle and not others within our cells. What's the name of the theory that explains that? Endosymbiotic. The endosymbiotic theory. Remember, endo means it's coming inside, and symbiosis is a relationship between 2 organisms. And you were taught those in high school, like predation, where one lives and one dies, or one benefits, one dies. Parasitism, one's benefited, one harmed. Mutual, they both benefit. Or commensalism, one benefits and one's unaffected. In this case, it was just engulfed. It was a relationship between the mitochondria and the bigger cell that engulfed it. Okay? What type of cells have mitochondria? Eukaryotes do. What are the 4 groups of eukaryotes? Fungi, animalia, protist, plants. Yes. Fungi, animalia, protist, and plants. They all have mitochondria. And those mitochondria take food and turn it into ATP. We already know that. But what about prokaryotes? Do they need energy? Yes, because all living things need energy. And you are 100% right. It is the cell membrane that metabolizes this process for a prokaryote. Because they don't have a mitochondria, they use their cell membrane to do so. Okay? What else did we talk about? Ooh, yeah. Your mom gives you your mitochondria. So in forensic studies, anytime that your nuclear DNA is disintegrated, because it will go first, we can always pull from your mitochondrial DNA, which is unique, but matches your mom. And so all of your maternal siblings will also have that mitochondria. Yeah, very good, very good. What else? I can't remember what else we talked about. Oh, yes, we talked about aerobic versus anaerobic. Difference? Aerobic, we have oxygen available. Anaerobic, we do not have oxygen available. Okay? Which one gives us the most amount of ATP? Aerobic, fantastic. And we're going to touch on that again today. Anaerobic gives you markedly less ATP. And that process would be called fermentation, but we will end on that today. Okay? What'd you say? Oh, yes. Which is going to take us into where I am today. Okay, good. So, we were talking about that we use food and we turn it into energy. And on that note, I said energy cannot be created or destroyed, but it can transform. It can go from being sunlight, and it can be changed and moved or transformed into chemical energy, so like food, and we can take that food and turn it into energy that you and I use. It's transformed. Now, there was a slide the last time we spoke that told you what actually allows for it to transform. How do we take sunlight and turn it into food? What is moved around? What is moved around? And sometimes we gain them. Electrons, which is taking me into redox, which is what we discussed last time. So, in order for energy to transform, electrons have to move around. Okay? And what better way to transport electrons than with electron carriers? What do you think electron carriers are responsible for doing? You guys. Yes. That's their one job. So when I initially started to study this, I was like, I have no idea. Like, what do you mean we have to carry electrons? So, of course, I'm trying to parallel it to something I already know. And these electron carriers, they pick up electrons and they take them somewhere and they drop them off. So then I was like, okay, maybe like a taxi. And let me just say, yesterday I used this example, and a student was like, I was like, you don't know what a taxi is? And she was like, is that kind of like Uber? Like, I don't think I'm that old. Like, I feel like you should know what a taxi is. But if you don't, that's okay. I'm going to use Uber for my example today because I was definitely humbled yesterday. I was like, what? Not a taxi. Okay. But what does an Uber driver do for its clients? It picks up that client and takes them to their destination and then drops them off. It picks them up, takes them to their destination, and then drops them off. Now, using the words that we used last week and substituting a client for an electron, when I pick up electrons, I need the two words we used last week. When I pick up electrons, I am reducing, I am gaining electrons. I'm going to carry them to my destination, and then I oxidize them. I'm going to drop them off. What's that Uber driver going to do next? Go right back, pick up more electrons, reduce, go to my destination, and oxidize. Guess what these electron carriers do all day long? Exactly that. They reduce themselves within their chemical reaction to pick up electrons, because when energy is released, it's released in the form of electrons. And so the only way for us to keep it is to catch the electrons. So if we're going to catch the electrons, we have to have an electron carrier there to catch it. In cellular respiration, we have two electron carriers. Two electron carriers. NAD plus. I read it NAD plus. I know it's NAD, but I always say NAD plus. And FAD. Within your notes right here, it tells you, if you want to say it out loud, what that's an acronym for. Okay? But you can totally get away with calling it NAD, and FAD. Now, these are my electron carriers. Oxidized form. This means that they do not have extra electrons attached to them. And let me clarify two things before I talk about their reduced form. Whenever we see electrons within our notes and our text, there's ways that they're represented that I want to make sure you understand that they truly just mean the same thing. You may see it says 2 electrons. So people normally see that electrons and they're like, okay, that's electrons have a negative charge. I get it, 2 electrons. It may say that, or it may say H or H plus. First of all, how many electrons does hydrogen have? 1. So does it make sense when it just has an H that they're like, oh, that's an electron? Because it's just got 1 electron. It does become confusing when you start looking at all of this stuff. You're like, oh my gosh. I don't know what the difference is between an electron and why do we have hydrogen ions here? Why do we have that? I need you to understand that at this level, when we're referencing electrons, it may look like this. Okay? Either one is acceptable. So I don't want you to look at these hydrogens and say, that's not electrons. Technically, hydrogen does have 1 electron. Okay? Also, we talked about the term phosphorylation. We talked about the term phosphorylation and what does it mean to phosphorylate? I have ADP and I'm going to make it into ATP. Is this getting bigger or smaller? How do you know? Diphosphate is 2 phosphates. Triphosphate is 3. So is this an anabolic or catabolic reaction? Anabolic because it's getting bigger. Fantastic. And when anabolic reactions take place, do we use dehydration or hydrolysis? Dehydration. We take water away to build things. Okay, I wanted to touch base on those 2 things before I went further into this. This was just a review, because that's what we're doing. This whole thing, we're making ATP, we're phosphorylating it, but I wanted you to also be able to recognize what electrons may look like. Now, back to my electron carriers. So, NAD plus is going to pick up electrons. When it picks up electrons, it goes from being oxidized to being reduced. What NAD plus looks like when it's been reduced is NADH. So this is its reduced form. When you see NADH, you know that that's carrying electrons right there. And when it gets to its destination, what's it going to do? It's going to oxidize them. It's going to let them go, and it's going to be this again. So this is my Uber driver. Here he is just waiting, or she is just waiting. Picks up somebody, takes them to their destination, and then drops them off. Like it just goes over and over and over again. The whole purpose of these electron carriers is nothing more than to carry electrons. FAD is my second electron carrier used in cellular respiration, and its reduced form is FADH2. FADH2. Both of these electron carriers carry electrons, which is why they're called electron carriers. But when you try to parallel them in your life, so what if this is like Uber and this is like Lyft? That's right, Lyft. I was like, oh, what is that other thing called? They both do the same thing, they're just different companies, okay? So you have Uber and Lyft, both carrying electrons, both carrying clients from their destination, or from their origination point to their destination, going back and forth. So you just have 2 different companies right here, okay? Picking up their electrons, becoming reduced. When they get there, they oxidize themselves to drop those electrons off. So electron carriers. We're going to discuss them as we go through the rest of our cellular respiration process. I'm going to move this right now. All right, may you go ahead and get your mitochondria out, please. Thank you. Maybe this way? Yeah. Okay, I am going to go back to here. Raise the screen up. I'm going to move it back down to center. Not exactly perfect, but it's pretty close. Okay, so what we talked about the last time, we started going through the details of cellular respiration, and I just wanted to give you a good base to start with. So we talked about the mitochondria in general. Then we talked about the 4 steps that are going to take place in the process of cellular respiration, the first one being glycolysis that occurs outside the mitochondria. Then the second, third, and fourth, pyruvate oxidation, the citric acid cycle, also called Krebs, and electron transport system, all happening inside the mitochondria, where conditions are aerobic, versus outside the mitochondria in the cytosol, where conditions are anaerobic. So you should already have all of this. I also gave you kind of like a foreshadowing to kind of to pre-teach before we got this far, how many ATP molecules were phosphorylated during each step. Whenever they're phosphorylated, what does that mean? They go from being ADP to ATP, which is an anabolic reaction. So I had us here. Now, I'm going to add details today, quite a bit of details. But when everything gets really fuzzy and you're like, I really don't know what you're talking about anymore. Big picture, what did I tell you is occurring here? Big picture with cellular respiration, what's occurring? We're using food, and we're using that food, and we're turning it into energy, yes. So when you're confused, and you're like, I don't even know why this even happens. How did I not even know? That line definitely isn't as straight as I thought it was. I just want you to remember, big picture, it's food to electrons. I also told you that it is probably very difficult to study with a human unless the human is studying the same material, because this is just not interesting to people if they don't have to know it. Okay? Your dog will be excited if you're like, oh, well, 2 molecules of NADH were generated. They'll be wagging their tails and excited. You have to be excited about it to be able to study it, to know it enough to pass the test. Okay? So big picture, food to energy. Now, what are we using as our food? What do you and I use for food when we're trying to make energy? Yeah, sugar. Specifically, which sugar? Glucose. Glucose, which gets us started on our first step here. Glycolysis. So our reactant is glucose, and I'll start with that here. Glucose is what goes in. Remember, reactants always go in. Products are what? Come out. Now let's talk a little bit more about this step before I move on to the products. The title of this step is glycolysis or glycolysis. How does that make sense in this situation? What does glycolysis mean? Glyco is sugar. Separation, lysis, or like the splitting of sugar. Glycolysis means sugar splitting when you interpret it. Now, Clydell, you just mentioned something and you said like break down. Does it make sense in step 1 that we're going to start breaking down glucose? Why does that make sense, big picture? I'm breaking down food to make energy. So it makes sense that I have to start with my food and start to break it down. So glycolysis makes perfect sense. I'm going to take that glucose and I'm going to start breaking it down. Now, there's a bunch that goes on here that you don't have to be familiar with, but once everything has taken place, let me tell you what you will be left with, which is what you do need to know. In step 1, you'll start with glucose. You will release 2 pyruvate molecules, 2 NADH, and then, of course, our 2 ATP. And I'm going to continue to explain. I'm just going to give you chances to write. So again, after we go through the series of reactions that take place here, that glucose is broken down, which makes sense, we're splitting sugar, into 2 pyruvate, 2 NADH molecules, and 2 ATP molecules are phosphorylated at this level. Now we've made this stuff. We have, because we've only made two units of energy right here. Is that going to be enough for you and I? Not even close. And I'll talk about quantities of energy in just a little bit. So I have to further continue this breakdown where I'm taking my food and I'm going to continue to break it down. These 2 pyruvate, I just want you to use common sense and kind of look at what's happening in the future. Where do you think these 2 pyruvate are going to be used? Why do you think it's going to be in the second step? Yeah, it's clearly called pyruvate oxidation. And with that being said, pyruvate oxidation, what am I doing to pyruvate? Oxidizing, so am I making it bigger or smaller? Smaller. I'm continuing to break it down. Okay, so these pyruvate are going to go to step 2. So I'm kind of putting it right here, the destination. The destination of that product. Bless you. Now, what are these NADH? What are those? Those are electron carriers. So when I split glucose, energy was released. And so I had to have something there to catch those electrons. Now, again, using logic and kind of looking what's going to happen in the future, where do you think I'm going to take those electrons? To step 4, the electron transport system. So I'm going to write a little 4 right here so you know that that's where they're going to be used next. And then I've phosphorylated 2 molecules of ATP. What am I going to do with that energy? I'm going to use it. Yeah, I'm going to use it. So I don't want to say that it's going to move this way because it's not. I've already created that. So this is going to be released. This will be used by the cell. Now, when I phosphorylated these 2 molecules of ATP, I used a substrate to do so. You and I have discussed substrates before, so sugar's a substrate, salt's a substrate, anything that you're trying to, like, there's in a solution, you have the substrate. So in this case, what do you think my substrate is? It's the sugar. So I'm going to put that out beside here. And basically when I say substrate level phosphorylation, because remember this is the type of phosphorylation, when I say substrate level, that means I used a substrate to make this, to phosphorylate. There was a substrate physically used to do that. Okay? So just to recap on step 1 real quick, my glucose came in, which was my food. Step 1 is called glycolysis or glycolysis, which literally means splitting sugar. And that makes sense because I'm going to need to break down that food in order to get energy from it. After a series of chemical reactions take place throughout glycolysis, I'm left with 2 pyruvate molecules that will be utilized in step 2. 2 molecules of NADH, these are my electron carriers that picked up those loose electrons, and they're going to carry them down to step 4. And then I phosphorylated at the substrate level, because a substrate was used, 2 molecules of ATP that my cell's going to go ahead and use. Okay, that was step 1. Step 2. Do you know what the reactant is for step 2? Yeah, it's the 2-pyruvate. It's the 2-pyruvate. So I'm going to kind of do this here. My 2-pyruvate go in. And through this step, no surprise, those pyruvates will be oxidized. And again, you don't need to know the specific details of that oxidation process, but once that oxidation process is over, the products are 2 molecules of acetyl coenzyme A, 2 NADH, and 2 CO2. Now, I don't think it's very straightforward as to where the acetyl coenzyme A is going to go, so I'm just going to tell you, but you could probably hypothesize. It's going to step 3. The 2 NADH, those electrons that were released, were harnessed. They were picked up. Where are they on their way to? They're on their way to step 4. Okay, now we have carbon dioxide. The process is cellular respiration. We're just releasing it. Yeah, it's carbon dioxide. We're releasing it. It's a waste. We took in oxygen. We're going to release carbon dioxide. Yeah. Fantastic. I love that you can already think that far ahead. Now, how much ATP was made here? None. This is the only step that no ATP is phosphorylated in. This is the only step that no ATP is phosphorylated in. All right? So we took those 2-pyruvate and we oxidized them. We made them smaller. We came out with 2 acetyl coenzyme A's, 2 NADH, and 2 CO2, which will be released. Okay? And when I go through the notes, if you're using eCampus, then I'll explain it, of course, on my notes. But I've already done your calculations for you, and I'll explain what I mean in a little bit. But so what I have here are your totals here. Okay, and I'll explain when I get to notes. So, what is my substrate, or what, well, it is my substrate, but what is my reactant for step 3? 2 acetyl coenzyme A molecules. Now, this citric acid cycle is truly a cycle, and it goes around and around and around and around. There's a lot that occurs here. You don't have to know all those details, but there is a lot that occurs there. So, after all 6 cycles have rolled through, what you're left with is 2 FADH2 molecules, 6 NADH molecules, 4 CO2, and 2 ATP. What is FADH2? It's an electron carrier, so where's this going? Step 4, right. What about NADH? Step 4. What do we know about CO2? It's out, it's released. And this ATP that was phosphorylated, it will also be utilized by that cell. Fantastic. Yes, it's released. Can you tell me why in step 3 we still refer to this as substrate level phosphorylation? was substrate level phosphorylation here in step 1. But why is it still substrate level phosphorylation in step 3? I'm using a substrate. I'm using something and further breaking it down in order to phosphorylate that ATP. So in this case, this acetyl coenzyme A is my substrate. Yes. Okay. Let's roll on to step 4. So ultimately what we've done so far is we started with glucose. We broke the glucose down to pyruvate. We broke the pyruvate further down to be acetyl coenzyme A. And now all we're left with are electron carriers. So the glucose has been broken down completely. Now we need to take all those electrons and use them for what they're for. So tell me what my reactants are here in step 4. I have 2 FADH2s. Yes? 10. How come I have 10? 6 plus 2 plus 2, which is 10. Fantastic. Now, before I go any further, I'm going to illustrate my electron transport system. What's happening truly? There's this little current that it's like the highway to step 4, because these are electron carriers, they're taxis, they're Ubers. They've made their way down to step 4. Can you tell me, now that the Uber drivers are here, these electron carriers are here, what are they about to do? Oxidize. They're about to drop off all these electrons. So they're all going to oxidize, and they're going to drop off tons of electrons. Tons of electrons are going to be dropped off here. And in this same location, there's this structure. And I will venture to say you can already tell me what it does. Now, this is what I think it looks like. I'll explain in just a second. But the name of this structure is called ATP synthase. ATP synthase. What do you know about this structure just by its name alone? How do you know it's an enzyme? It ends in ase. And if it's an enzyme, it has to be a protein. All enzymes are proteins that, what do they do? Enzymes are proteins that speed up reactions. Enzymes are proteins that speed up reactions. What reaction is ATP synthase carrying out? ATP synthase. What is synthesis again? Making or creating. So ATP synthase means I'm making or creating ATP. This enzyme is responsible for speeding up the reaction of making ATP. ATP synthase. Just its name alone, if you can analyze it and see that the ACE makes it an enzyme and all enzymes are proteins, you know that if it's an enzyme, it's responsible for speeding up a reaction. ATP synthase, it's speeding up the reaction of taking ADP and turning it into ATP. Fantastic. Let me tell you what this looks like. And some of you are going to make fun of me. Nothing new. Okay, so you tell me that there's this protein down here, and it's making ATP. Okay, it makes sense because it's on a diagram, and I've drawn it there. But I wanted to know, what does it really look like? So I started searching YouTube. And this protein spins like it constantly spins. So I'm watching all these videos about it, and it's spinning, and I start trying to parallel it to something I already know. And the first thing that came to my mind, and I don't know what it's called, but you know on the roof how you have those metal things that, what is it called? Vents? Is it called, like where it spins? Yeah, I called them a wind turbine. I didn't know. Okay, I was just not really sure. So that's what I've called them. And you're the very first person who's corrected, so I'm grateful for that. It just spins super fast, especially when the wind's blowing. It's like, that's what this thing here looks like. And because it's spinning so fast, which is what those turbines do, they create a current pulling all the heat or all, whatever it is that it's trying to release from your house. So it's spinning and it creates this current. So it's pulling all these electron carriers down here. It's just spinning, and it's created this current. The electron carriers truly go through here because it's a channel. It's got a lumen. It goes through here super fast, and it's oxidizing as it goes through, and it's shooting off electrons like this, like. And at the same time, you have ATP going like this. So it's like.
So those what's working, the energy.
Fantastic, and that's even going to be a test question. So if you took a medication that inhibited ATP synthase, how would it impact you? You'd be really, really tired. Really, your ATP levels would be really low, and it could lead to your expiration. Yes, 100%. So this right here continues to spin. And as these electron carriers go through here, it shoots off those electrons. It's oxidizing them, and it's phosphorylating ATP at the same time. So there's just like crazy fast. So we've oxidized these electrons using ATP synthase, and through this process we've generated 32, roughly, this is a range, 34 ATP in just step 4. Can you tell me why at this point we call it oxidative phosphorylation and not substrate level? 100%. I had no substrate. I just oxidized these electron carriers. So it's called oxidation phosphorylation. I still phosphorylated, I still got ATP, but in this case I didn't have a substrate, I used electrons. I just oxidized them. Just generally speaking, this process that we have up here, roughly how many ATP were phosphorylated? 36 to 38. And I'm going to tell you, that says 32. 36 to 38. I'm going to tell you that it's always going to be a range. And what I mean by that is there's no definite number of ATP that are phosphorylated each time. And some people are really efficient at doing this and produce more ATP, and some of us aren't, and we produce less. Now, in our example here, it says 36 to 38. Depending on what textbook you use, they may say there's like 30 to 34. So I need you to understand there's no, it makes 36 ATP. There's none of that. It's a range. And that just has to do with the way the electron carriers work and the efficiency of your individual cells, which is unique to you. Some of us are really good at it, and some of us aren't. And not that you can train at it, but it's just a genetic thing. Now, if you currently don't have a lot of ATP, perhaps because of your lifestyle, can you change the amount of ATP? Absolutely. You can increase your activity level, and we discussed that in the previous lecture. And your mitochondria, once they're maxed out, they'll create more through a process of binary fission, which is how bacteria divide, which gives us more evidence that mitochondria used to be bacteria. But they'll create more mitochondria, so you can make more ATP, because your activity level is higher. Did I answer your question?
So does each cell is different ATP, or is the whole body of the cell that's
Exactly what I was about to talk about. Fantastic segue. So, in your body, let's say you have about 100 trillion cells. I'm going to turn this light off real quick, just for a moment, so that there's a little bit more contrast. 100 trillion cells in your body. And let's say for the average activity level, you have about 6 mitochondria per cell. So you're already, we're making this every half second in 1 mitochondria. So every second, your range is doubled. So let's say it's 72. You're making 72 every second per mitochondria in each side, inside each cell. Now, if that's just the average, for people who are extremely athletic or have a lot of endurance built up, they don't have just 6 mitochondria. How many are you thinking? 12, 15. Okay, you can increase based on the levels of ATP you need. That's why when we talked about working out, you can't just go from 0 to 100. Once you start to do it, you'll build up. You'll create more mitochondria through the process of binary fission, and as a result, you'll generate more ATP. Say you build up a whole lot of mitochondria and a whole lot of ATP, and you say, well, you know what, I'm not doing this anymore. I'm not going to do all this activity for the rest of my life. I just will stop. Do you know what your body does with all those extra ones? Doesn't reuse them. If you don't use it, you lose it. Can you tell me what organelle will go in it? Lysosome. Get rid of it. Hey, we needed you at one point. Your services are no longer needed. And you're out. The lysosome gets rid of you. Because it's a lot of work to maintain high energy levels. And those of us who do use a lot of energy, and I didn't use those of us like I said to me, but like those people who do use a whole lot of energy, they require a lot of resources. What do I mean by resources? Food. A lot of food. When you have bodybuilders and people that are doing that kind of stuff, they don't eat your 1,500 calorie diet. They're eating like 5,000 and 7,000 calories a day. They need more energy, so they need more resources. Okay? So that's this process here happening exponentially in your body, like zillions per second you're generating. Now, what if you die? What if you die? Well, I mean, at one point you're going to die, but what's going to happen there? Okay? So, do I do aerobic or anaerobic? It's anaerobic because you don't have oxygen. And you'll continue to do this for a few hours. But then after everything's been depleted, the mitochondria can no longer generate muscle movement. Can you tell me what your muscles are going to do as a result of no oxygen for an extended period of time? They become stiff. What do we call that process? You hear it in like medical exams and they're like, oh, this is set in. Rigor mortis. Rigor mortis is directly tied to the mitochondria. When we undergo anaerobic conditions, you and I are going to produce lactic acid. That's the very last slide I'm going to show you today. And it gets stuck in our muscles. And the longer we go without it, the stiffer our muscles become. And so we can pretty much give a pretty good time frame of death based on how stiff their muscles are. Because if they're fresh, their muscles are still they're dead weight, but they're still pretty movable. If they've been there for 8 to 10 hours, you'll notice that their muscles will start to, because there's no ATP anymore for them to do that. So rigor mortis is a good indication of how long they've gone without ATP. Questions on this so far? Okay, so I put pyruvate oxidate. They pair these two together because there's no ATP phosphorylated here, so they don't count this as a separate step. I teach it to you like that to keep it methodical, because if I'm like, hey, these are joined together, when I try to teach it like that, more people were unsuccessful at learning the process. So I break it down. She is 100% right. Melissa was saying in the book it says that there are 3 steps. They combine these 2 steps. Okay? I separate them because it's easier to memorize. You won't miss a question because it says 3 versus 4. I just want to make sure that you understand the progression of that cycle. Or that process. I don't want to say cycle. The process. That's an excellent point. Additional things that I can clarify before we move on. Okay. So, go ahead.
Cellular synthesis, where do you get the 32 to 34?
This right here?
Yes.
So remember when I said there's a complex series of reactions that take place? But ultimately, we're oxidizing all of these. And I'm going to tell you how to calculate how many ATP are generated per electron carrier in just a couple slides. So you'll be able to calculate. Like, I don't just pull this out of anywhere. There's like a mathematical calculation for it that you don't have to necessarily understand, but at least you see where I'm coming from on those numbers. Good question. Okay, I'm going to drop the screen. I'm going to leave that here so that afterwards, when we're done, if you want to take a picture or whatever, you're welcome to do so. Okay, so this is where I left off on the last day we were together. I talked about the 4 stages, and even in your notes we've taught it this way. But I'm really grateful, Melissa, that you brought that up. And again, if it talks about 3 stages, it combines these 2. That's all that is. I just separate them to help you with the reactants and products. I started today with what my electron carriers were, so you are familiar with those. Step 1, glycolysis. These match what you have in eCampus. I've just reformatted them to where, because yours is divided up where one slide has the reactants, one slide has the products, I put them together. So my reactants are on my left, my products are on my right. My reactant in glycolysis is glucose, and my product in glycolysis is 2 ATP, 2 NADH, and 2 pyruvates. I'm not going to go through all the steps and everything, but pointing out 2 things. It says it occurs in the cytoplasm, which you are familiar with. Everything I've taught you here, I am reading out here. It's provided for you in words here. Cytoplasm is the location, and glycolysis literally means sugar splitting. Okay? Here's the diagram in your textbook on glycolysis. I just want to say a caveat really quickly. As you go through this and you memorize it, if you do further go into biology, specifically cellular biology, when you become a junior or senior, you're going to take really complex science classes, and there's one that's just focused on the mitochondria, and then you can understand these steps and the complexity that they're introduced. But where you're at right now, which is what we call a 1,000 level class, it starts with a 1, you do not need to know the steps in that much detail at that level. Okay? You just need to know the location, the conditions, the reactants, the products, and the level of phosphorylation, which is exactly how I taught it to you. But we include these diagrams because these are the diagrams that are used in your book. I did talk about what it meant to be substrate low, to be phosphorylated at the substrate level, which means that you had some type of substrate. In this case, it was glucose to begin with, and then it was acetyl coenzyme A in step 3. Step 2, pyruvate oxidation. I did the same thing with the notes. My reactants on the left, my products on the right. Now, when I was drawing on the board, I mentioned I already did your calculations for you. What I meant by that is, right here, it tells you that pyruvate is converted. It's referencing 1 pyruvate, but how many pyruvate went in? 2. For each pyruvate, you will have 1 acetyl coenzyme A, so 1 acetyl group. So I put 2, right there, 2. For each one, you'll have 1 NADH. So I put 2, okay? So you understand what I'm saying here. I did the calculations there so that we weren't going back and correcting and you weren't confused in your notes, all right? Here is the citric acid cycle, or the pyruvate oxidation. Of course, this is citric acid, but pyruvate oxidation happens right before that. Again, if it says that there's only 3 steps, it truly combines these 2. And they're both shown in all of the diagrams. There's not just a diagram of pyruvate oxidation on its own. Step 3, the citric acid. Again, I have acetyl coenzyme A going in. as my reactant, but remember I had 2 molecules of that. My products per acetyl group were 2 CO2, 1 ATP, 3 NADH, and 1 FADH2. So I had done the math already. That's why I said we had 4 CO2, 2 FADH, 6 NADH, and 2 ATP. This is the citric acid cycle, also called Krebs, again with a capital K because it was named after a scientist, but you do not need to know it at this level. This is the diagram straight out of your book. It's cool if you want to study it, but I guarantee, unless you're like passionate about it, you don't want to study it. Step 4, the very last one. I don't have any specific substrate. All I have is a bunch of electron carriers. They've made their way to step 4. And what's going to happen to them at step 4? They're going to oxidize themselves. They're going to drop all those electrons off. And there's going to be a protein there called ATP synthase that's going to take ADP and turn it into ATP. And it mentions that there as you kind of go through and read it. Big picture, though. Oops. Okay? This is the only diagram that kind of mimics what I have down here. It's still a little, obviously it's different than mine, but it does give you a pretty good idea if you don't understand what we drew together out of your text. Now, here's what Andrea was asking about. Where do you get the 32 to 34? How did you come up with that number? Did you just pull it out of the air? The answer is no. What this is showing you that is in aerobic conditions, which means I have adequate access to oxygen, for each NADH molecule, I can synthesize anywhere from 2.5 to 3 ATP molecules. So I had 10 NADH at the end, so 10 times 2.5. Okay, so I could get anywhere from 25 to 30 ATP just from the NADH. From my FADH2, I had 2 of those, and each of those can give me anywhere from 1.5 to 2. So then again, you have 3 to 4. And that's that range I'm talking about. There's no specific, this is how many are generated every time. It's a range, depending on the efficiency. And these ATP here are in addition to the 4 that were phosphorylated at the substrate level. Okay? So let's just say we were at the high end here, so 30 plus 4, right? So I'm using only my big numbers, so that's 34 here, plus 2 here, plus 2 here. So I'm at 38. I'm at 38. Now if I was on the low, do you see what I'm doing there? I'm creating that range. So that's how we calculate those. This diagram here is also in your text, and it shows you how they're calculated. It kind of, but it used the low number and gave me 32. So you can use anywhere within that range. Questions there? Yes, ma'am. Do we need to know that? I don't believe so. Okay, last topic. Fermentation. Fermentation takes place in anaerobic conditions, which means what? I don't have any oxygen. I'm not breathing. Okay? Now, you and I can't do this for a very long time because, of course, then we would expire. But what are some activities that may cause us to undergo fermentation? Swimming, sprinting, weightlifting. Okay? Anything that's high intensity that doesn't give us adequate access to oxygen. Now, for those of you who do those type of activities on a regular basis, your goal when you work out is to leave sore. Why is your goal to leave sore when you work out? Because you know at that point, you've pushed yourself. If you do a workout and you're like, man, that was a great workout, but the next day you're perfectly fine, did you really strain your muscles? No, you didn't. And scientifically, we can explain that if you do a high-intensity workout under anaerobic conditions, because you're an animal, what steps do you carry out? What steps of cellular respiration do you carry out under anaerobic conditions? Just step one. Step one. How many ATP molecules are generated there? 2, which is a huge difference from 38. So tell me how you feel after a few seconds of just making 2 versus 38 per half second. You're tired. You're tired. And because your muscles had to look somewhere else for resources in order to undergo a respiration, they have left you waste in the form of lactic acid in your muscles. Lactic acid is the waste product of this process, fermentation. And whenever that lactic acid sits in your muscles, at first you don't feel it. But once you stop moving for about 30, 45 minutes, deep couch sit, or you go to bed, when you wake up the next morning, you feel tired. You feel sore. And if you've worked out that hard, that's awesome, first of all. Way to go. Second, what a lot of people do is when they feel that soreness, what do they say? Yeah, you know what? I'm going to skip workout today. I'm still sore. But what should you actually do? You should actually work out, because the only way to get that lactic acid out of your muscles is to pump blood through it. And if you don't move, it's just going to sit there, and you're just going to stay sore. So it will eventually wash its way out after about 3 or 4 days. But if you really want that soreness to be alleviated quickly, you get yourself back in there and you do like a jog or a power walk and get that blood pumping through. So when you and I undergo anaerobic conditions, we carry out fermentation. Of course, we cannot do this for hours on end. This is in very short spurts. This applies to fungi, bacteria, and animals. Of course, bacteria and fungi do not have muscles, so they don't have, that's why the lactic acid, as far as muscle soreness, only applies to us. But they do produce lactate or lactic acid. Now, what you and I are going to study in lab is the fermentation that occurs in yeast cells, which explains how we make breads and beers. We will use fermentation chambers over there on the side, and we're going to set up 3 trials of yeast, each group will, and you're going to feed those yeast different things. And based on what they like, the more that you feed them, the more waste they'll make. And their waste is CO2, and that's what causes bread to rise or beer to ferment, to age for beer. Okay? So alcoholic fermentation is carried out by yeast cells. It's carried out by yeast cells. You and I do not carry out alcoholic fermentation. All right. On this diagram right here, let me just explain what it shows. That way, when you look back at it, you'll understand. Everything goes through glycolysis, and then it gets broken down into pyruvate. And then pyruvate, if we have aerobic conditions, pyruvate will go into pyruvate oxidation, citric acid cycle, and the electron transport. But if there's no oxygen present, it immediately goes through fermentation. And fermentation is the process that takes place under anaerobic conditions. And that's where I will end today. Is there anything that I can explain or clarify? All right. You and I are finished.